All right, this time we're going to focus on oxidative phosphorylation and the third step and the conversion of FADH2 and NADH captured in glycolysis citric acid cycle to ATP. And this is going to require oxygen, oxidative phosphorylation, and it's going to be the phosphorylation of ADP to produce ATP. And the components of that are going to be in the inner membrane of the mitochondria. So FADH and NADH2 will be in here and will be recycled in this process. Okay, and this is what it looks like. Let me just draw up here the mitochondria. And we're looking now at the phospholipid bilayer of the inner membrane, like that. And then this is the matrix. The light yellow is the matrix. And the orange is the region between the inner membrane and the outer membrane. So it's a small region, and it's a special region. And look what happens in this special region. What happens is that NADH and FADH2 feed into something called the electron transport chain. And as the electrons that they give up pass through the electron transport chain, these protein complexes, the big purple globs floating in the inner membrane, actually pump hydrogen into this space between the inner and outer membrane. And this space becomes, this orange space becomes acidic. So we've got a proton transport pump in the inner membrane, and it's driven, by the energy released by NADH, FADH2. Why this unusual process? Because it's a very efficient engine. So remember we talked in a couple of mini lectures ago about the idea of engines and the efficiency of engines and their capturing of energy. So your car engine is like your body engine. And this happens to be a very efficient way of capturing energy. So what we're going to do is we're going to first build up a high concentration of hydrogen ions in this region, and acidic pH in this region. And then over here, another important protein in the inner membrane is going to grab it, and this is an enzyme that catalyzes the synthesis of ATP and uses this hydrogen ion as a proton motive force. This process is called chemiosmosis because it's similar to osmosis, letting water through, but in this case we're letting a hydrogen ion through. So it's called chemiosmosis, and that's the driving force for the synthesis of all the ATP. And so the bottom line is we're converting NADH to FADH2 using the electron transport chain plus chemiosmosis. The overall process is called oxidative phosphorylation. And here's a really nice, I think the next one is a really nice view of it, because this puts it on that energy diagram, where we're going to tumble down through this electron transport chain that has several different components to it. And in some steps, there's enough of an energy drop that we can pump ions across the membrane. Look how they feed in. NADH feeds in at a higher level than FADH2. And so that means more protons can be pumped and ultimately more ATP. So in the case of NADH, we can get 3 ATP out of the deal. In the case of FADH2, since it's a lower energy molecule, we get 2 ATP out of the deal for it. Okay, like that. And the ultimate bottom of this electron transport chain accepts these electrons, right? So we've got actual electrons being handed off in redox chemistry. Molecules are getting oxidized and reduced as they go tumbling down. And the final electron receptor is oxygen. So this is why big reason why we're breathing in oxygen, and that oxygen is getting converted to H2O. So significantly, the CO2 we breathe out, that oxygen is not the same oxygen atoms as the oxygen we breathe in. The oxygen atoms we breathe in just gets converted into some water, H2O, in our bodies, like that. And so the CO2 that we're breathing out, the oxygen is really from the foods we ate. the carbon and oxygen are from foods we ate. And so you need oxygen for the electron transport chain to occur. And the next thing to say about that is there's a focus on ATP synthase. So here it is, it's a protein. It's got quaternary structure. It's actually multi-protein complex that's getting this job done. But we could zoom in like we zoomed in on aquaporin, and we could look at this in detail, and we could spend our whole course studying the primary amino acid sequence, secondary structure, tertiary structure, and quaternary structure of ATP synthase to see how it can use this proton motive force in chemiosmosis to drive the synthesis, right? So we've got what in this case is not a chemical reaction of dropping an exergonic reaction. It's really a question of diffusion, and we actually started out the first mini lectures in Chapter 8 talking about the concept that concentrations of molecules across a membrane provide one type of potential energy. And here we are harnessing some potential energy and converting it into chemical energy using this ATP synthase. So a pretty impressive protein enzyme connecting these two reactions. And that's actually all we want to say about the oxidative phosphorylation, except to note that this is where we get the bulk of the ATP Maine. And then the final mini lecture from this is going to talk about what about in situations where we run out of oxygen. And when we run out of oxygen, we can't do oxidative phosphorylation, and everything backs up, just like a big traffic jam. These chemical reactions all back up, and we have lots of NADH and FADH2, and so things stop. The citric acid cycle stops because there isn't enough FADH and NAD plus to allow the reactions to continue. And we'll see in the fourth mini lecture from Chapter 9 that the book ends Chapter 9 in a example of a metabolic pathway called fermentation. And that's that.